Right, and proceeding from the chromosome assembly, we are going on across the subject matter which was discussed a few minutes earlier. And from this, with just a general mention, I'll be going on to the next topic. Here, the DNA binds to start the assembly of the enzymatic replication machine. The early stages of this process involve the assembly of a chromosome, right? And that functions to unwind two strands of DNA at the replication forks and add RNA primers to the DNA template. Templates that will be used by the DNA polymerase enzymes to begin replication. And subsequent to the remodeling of the replication origin induced by DNA-A, the assembly of the bacterial loader-dependent chromosome occurs in discrete steps and involves four different proteins: initiator protein, helicase, helicase loader protein, and primase that act in a coordinated and sequential manner. The ORIC-C region of prokaryotes contain highly conserved sequence motifs that include an AT-rich box domain that serves as the recognition sequence for the binding of the DNA initiator protein. Initial binding of DNA-A to ORIC-C promotes the melting of the DNA double-helix and the recruitment of multiple DNA-A subunits that form a helical oligomer along the newly opened single-stranded DNA, ssDNA. The DNA-A protein contains four major domains. The domains 3 and 4 are integral to binding the ssDNA, while domain 1 is involved with protein-protein interactions. Domain 2 forms a flexible linker between the protein interaction domain and the domain binding domains. This is the picture, the illustration of that, DNA melting and helicase loading at ORIC-C. It's already been shown to you, but just for a recap. ssDNA, domain 3, domain 4, and this is the separate picture of that. Here is the direction of the DNA movement during the replication. Take screenshots of these and it should be very helpful to you. All right. And going from this, we'll go into the subject matter which we stopped at the last lecture. The E. coli system, the helicase loader protein, DNA-C complex with ATP binds to hexameric helicase DNA-B and forms a DNA-B DNA-C complex, which has been confirmed by cryo-electron microscope AIIMS studies. The loader protein delivers the helicase onto the melted DNA single strands of the DNA-A ORIC-C nucleoprotein complex at the origin of replication in vivo. This delivery is associated with the initiator protein, DNA-A, whose amino terminal domain NTD is thought to have a role in loading the helicase and helicase loader complex onto the ORIC-C by interacting with helicase. DNA-B. After the loader protein dissociates from the helicase ring, the functional chromosome within the chromosome, let me repeat that. After the loader protein dissociates from the helicase ring, the NTD of the helicase interacts with the carboxy terminal domain, CTD, of the primase and forms a functional chromosome. Within the chromosome, the helicase DNA-B acts to unwind the double-stranded helix, and the primase DNA-G synthesizes RNA primers on both the leading and lagging DNA strands. Excuse me. Helicases are motor proteins that move directionally along a nucleic acid phosphodiester backbone, separating two annealed nucleic acid strands such as DNA and RNA using energy from ATP hydrolysis. There are many helicases representing the great variety of processes in which strand separation must be catalyzed. Approximately 1% of eukaryotic genes code for helicases. The human genome codes for about 95 non-redundant helicases of 64 RNA helicases. And 31, DNA helicases. Many cellular processes such as DNA replication, transcription, translation, recombination, DNA repair, and ribosome biogenesis involve the separation of nucleic acid strands that necessitates the use of helicases. In E. coli, the DNAB helicase as seen in figure 9.5, is responsible for unwinding the two parent DNA strands to unwind and separating them from one another to form a Y-shaped replication fork. The replication forks are the usual site for DNA copying. During replication within the fork, helix destabilizing proteins called single-stranded binding proteins, SSB, bind to the single-stranded regions, preventing the strands from rejoining. DNA polymerases. DNA polymerase enzymes are required for the assembly of the daughter strands along each of the template DNA strands. All DNA polymerases require a DNA template and a primer that is used to begin the replication process. The primer is a short strand of RNA that is replaced on the DNA template by the Primase enzyme. Recall also that DNA contains two antiparallel strands and that DNA polymerases can only add new nucleotides in the 5' to 3' direction when synthesizing the daughter strands of DNA. Since both strands of DNA are replicated simultaneously by the same replisome, the leading strand, where the daughter strand of DNA is moving in the 5' to 3' direction, is replicated continuously and flows in the same direction as replisome movement. The lagging strand that lies in the antiparallel direction has to be synthesized in the opposite direction of replisome movement and is created using short bursts of DNA polymerase activity, leading to the formation of Okazaki fragments along the template strand. Thus, the lagging Okazaki fragments, lagging strand must continually be primed with short RNA sequences to maintain the formation of the Okazaki fragments. The RNA primer sequences must then be replaced by DNA, and gaps in the backbone of the DNA must also be repaired. E. coli has a total of 5 DNA polymerases. 3 of these enzymes are involved in DNA replication, DNA polymerases 1, 2, and 3. The DNA polymerase 3 is the main polymerase involved in both leading strand biosynthesis and synthesis of the Okazaki fragments during DNA replication. The DNA polymerase 3 holoenzymes is comprised of 10 different proteins organized into 3 functionally distinct but physically interconnected assemblies. 1. The alpha epsilon theta core. 2. The beta sliding clamp. And 3. The delta TNY3N delta psi X clamp. That's pretty numerical, figurative, and a lot of indicators there. Clamp loader complex, figure 9.6. Okay. In the polymerase core, alpha is the polymerase subunit, epsilon the 3' to 5' proliferating exonuclease, and theta is a small subunit that stabilizes epsilon. After an RNA primer is made by DNA G, the B2 clamp is loaded onto the primer terminus by the clamp loader. The alpha and epsilon units separately bind the clamp, each via a short linear clamp binding motif CBM to the 2 symmetrically leading CBM binding pockets of beta 2. Tethered to the clamp, pole 3 is able to synthesize DNA at high speed, squabble 1000 nt per second, and with much higher processivity greater than 150 kb. This is the diagram for that. Just note the diagram. It could be a great help to you, right? And take a screenshot and enumerate on that. The entire details are there, where you have the pole 3 core, the clamp loader, lagging strand, and then the leading strand, and related details. Now, figure 9.6, the DNA replisome. The standard textbook model of a DNA repliposome showing the coupled and highly coordinated processes of leading strand and lagging strand synthesis. DNA polymerase 3 is connected to the DNA-B helicase through the T subunit. Is that the T? I hope I'm reading that right, because that's Greek letter, of the clamp loader couples, and 2 or 3 polymerase cores replicate DNA from both leading strand and lagging strand DNA templates concurrently. The ssDNA in the lagging strand loop is bound by the ssDNA binding protein, SSB. Recent studies have shown that E. coli DNA polymerase 3 is readily exchangeable at the fork, and that leading strand and lagging strand synthesis may not be tightly coupled or may even be accomplished by different DNA polymerase 3 holoenzymes. The DNA-B helicase can also be decoupled from the DNA polymerase couple complex and translocate ahead of the apex of the fork. Bacterial repliposomes are highly flexible and mobile machines, their dynamics being mediated and controlled by a network of protein-protein interactions at different strengths. Many of the replication proteins are either conformationally flexible or contain flexible unstructured regions, making structural studies by X-ray crystallography or NMR difficult. However, through decades of effort, structures of all E. coli replication proteins or their bacterial homologs have been solved as complexes, whole proteins or domains. Recent breakthroughs in single-particle cryo-electron microscopy (cryo-EM) have seen structures determined of large repliposome subassemblies, even the whole bacteriophage T7 repliposome, though so far only at modest resolution. Cryo-EM structures of the E. coli Pol3 co-clamp TC C-terminal domain of the T subunit of the clamp loader complexes and prime template DNA in both polymerization and proofreading models were recently solved at 8 and 6.7 angstrom respectively, along with structures of a DNA-free complex, as seen in Figure 9.7. These structures resemble previously proposed structural models with surprises as well. For example, in the DNA-bound polymerization complex, the beta-2 clamp becomes almost perpendicular to the DNA strands, as in 9.7 A and B, in contrast to its tilted configuration in the crystal structure of DNA-bound B2, while the Pol3 alpha polymerase subunit binds to DNA in a conformation similar to the crystal structure of DNA-bound Thermus aquaticus alpha. The locations of C-terminal domains, alpha CTD, comprising the oligonucleotide binding domain OB and the T-binding TBD domains, are different. In the TAC alpha structure and the DNA-free complex, the alpha CTD is close to the polymerase active site with OB domain positioned to bind and deliver the ssDNA template into the active site. Figure 9.7c and D. In the DNA-bound cryo-EM structures, these domains are shifted towards the little finger domain of alpha, the domain that directly contacts the beta-2 clamp. They are therefore far away from the template strand, entering the active site, Figure 9.7e. The OB domain contacts both the little finger and thumb finger domains of alpha as well as the beta-2 clamp and epsilon. The phase of the OB domain that had been thought to be involved in ssDNA template binding now directly faces and is relatively close to the dsDNA. Additionally, epsilon wedges between the alpha thumb domain and the clamp. This previously unappreciated interaction network apparently stabilizes the whole complex. Have a look at this complex, this colored illustration. Take a transcript of it, a screenshot, and make your copies and do justice to yourself by getting enough explanations on that. Well, the explanation of the figure of each case that's shown here are highly helpful, so please go on with that. I'll just read it out for you anyhow. The structure of the E. coli polymerase clamp TC, I read it as TC, DNA complex is surface, the representation of the polymerization left, and proofreading right complexes. The N-terminal domains of alpha NTD, residues 196, are colored in deep salmon, and the OB 964, 1072, and T binding domains, TBD, 1173 to 1160 of alpha CTD in brown and dark salmon respectively, epsilon in yellow, beta in aquamarine, theta in orange, and TC in slate. So you can see all that here. Various colors and how it's done, right? So a pretty good illustration there. Make a color copy if you can. Of course, screenshots would be of great help to you. That being said, let's go to the next sentence. That is, the polymerization complex does not include theta and TC, and the alpha CTD are missing from the proofreading complex. Beta. Cartoon representations of complex left. The DNA has B-form structure, while in the proofreading complex, the primer DNA is frayed with the end of the newly synthesized strand at the active center of epsilon. The proofreading complex is rotated slightly to show DNA in the active center of epsilon, and the theta subunit is omitted for clarity. C. Surface representation of alpha NTD from the DNA-bound polymerization complex PDB FKB, showing the thumb, palm, finger, and PHP domains. Right? That abbreviation should be clear. 5 FKB. Okay. And D. Positioning of the alpha CTD in the DNA-free complexes. PDB 5 FKU. E. Positioning of the alpha CTD in the DNA-bound polymerization complex, PDB 5 FKB. While the OB domain in the DNA-free complex is close to the active site of pol 3 alpha, it is far away in the DNA-bound complex. The OB domain is colored in marine and the TBD in magenta. The alpha NTD gray in the two complexes shows relatively minor changes compared to alpha CTD. Now on to the subject after the figure explanation. Overall, there is a significant conformational change in the DNA polymerase. Right, and to move from interacting with the clamp in the DNA bound state to a position 35 angstroms away from the clamp in the DNA free state, as shown in video 9.1, it has been hypothesized that this large conformational change may help the polymerase act as a switch to facilitate the lagging strand synthesis. On the lagging strand, the polymerase repositions to a newly primed site every 100,000 BP. To do so, the polymerase needs to release both clamp and DNA. The switch-like movement of the polymerase tail may play a part in the release and consequent repositioning of the polymerase at the end of the Okazaki fragment. Video 9.1. DNA binding induces large conformational changes in the DNA polymerase tail complex. The video shows the linear morphing of the DNA free to the DNA bound state, showing the large conformational change between the two states. The green subunit is the beta clamp. The alpha subunit is shown in orange, with the active site residues in magenta. The alpha C-terminal domain to CTD is shown in brown, the alpha subunit in yellow, and the T-tol shown in blue. The proofreading complex is fairly similar to the polymerization complex, with the small movements of individual protein components. The most significant movements include a rotation and a tilt of duplex DNA against the plane of B2, locking the DNA against the inner surface of the B2 ring. The polymerase thumb domain and epsilon also move towards the DNA. The thumb domain wedges between the two DNA strands with unmatched base pairs, resulting in a highly distorted and frayed DNA substrate. The newly synthesized strand is therefore able to reach the nucleus active site of epsilon for editing. Considering that the proofreading complex is fairly similar to the polymerization complexes and duplex DNA with two unmatched base pairs tends to fray, it is proposed that epsilon works passively by waiting for DNA to reach its nucleus active center when a wrong nucleotide is incorporated rather than responding actively to the misincorporation event. In a complementary single-molecule biophysical study, the clamp-bound pol 3 core has been shown to be remarkably stable and processive in the proofreading mode in the absence of incoming dNTPs. Bacterial replisomes have long been believed to be highly coordinated, highly processive machines capable of copying the whole chromosome without dissociation. 2 or 3 polymerase cores of the same E. coli pol 3 She were believed to synthesize both DNA strands, with the lagging strand polymerase repeatedly being cycled for new Okazaki fragment synthesis, as described above. The lagging strand polymerase recycling has been debated to be triggered by various collision or signaling mechanism in a well-controlled manner that likely involves the movement of the T-tail region. However, recent studies find that bacterial polymerases also readily exchange replication forks, and that leading strand and lagging strand DNA synthesis may not always be tightly coupled. Figure 9.6b depicts a proposed model for this exchange. Overall, DNA polymerase 3 is a highly processive enzyme incorporating 600 to 1,000 bases per second, with greater than 100,000 bases incorporated per binding event and an error rate of approximately 1 per million. DNA polymerase 1 aids in the process of lagging strand synthesis in that this polymerase removes the RNA primers and incorporates DNA in its place. DNA polymerase 2, although not well understood, is thought to play an editing role following lagging strand synthesis by DNA polymerase 1. DNA polymerases 1 and 2 also play a role in DNA repair, as do DNA polymerases 4 and 5. DNA polymerase is similar to DNA polymerase 1, right? It's similar to DNA polymerase 3 in that it has 5' to 3' polymerase activity, and also has 3' to 5' exonuclease activity to mediate both the processivity and the DNA proofreading function of the enzyme. In addition, DNA polymerase 1 also contains a large protein domain called the clino fragment that exhibits 5' to 3' exonuclease activity. The 5' to 3' exonuclease activity is responsible for removal of the RNA primers along the lagging strand. The processivity of this enzyme enables the polymerase to refill these gaps with DNA. However, DNA polymerase is unable to connect with the backbone of the newly synthesized Okazaki fragments, right? And with the downstream fragment, the repair of gaps in the DNA backbone is mediated by a DNA ligase enzyme. This is the diagram of synthesis, proofreading, and primer removal. Here, the structure of the E. coli DNA polymerase 1 is a figure shown here. So DNA polymerase 1 exhibits 5' to 3' polymerase activity and 3' to 5' exonuclease activity, mediating the processivity and the proofreading activities of the enzyme. DNA polymerase 1 also contains a 5' to 3' exonuclease activity housed in a special domain of the enzymes called the clino fragment. Excuse me. And this domain is responsible for removing the RNA primer sequences from newly synthesized DNA. The 5' to 3' polymerase activity is then used to replace the RNA primer with DNA. The 3' to 5' exonuclease activity ensures that the correct bases are incorporated. DNA ligase enzymes seal the breaks in the backbone of DNA that are caused during DNA replication, DNA damage, or during the DNA repair process. The biochemical activity of DNA ligases results in the sealing of breaks between 5' phosphate and 3' hydroxyl terminal within a strand of DNA. DNA ligases have been differentiated as being ATP dependent or NAD plus dependent, depending on the cofactor for co-substrate that is used during their reaction. Typically, more than one type of DNA ligase is found within an organism, as shown in the figure 9.9. The DNA ligase enzyme is covalently modified by the addition of the AMP moiety to a lysine residue on the enzyme. The AMP is derived from the ATP or NADH cofactor. The downstream 5' phosphate at the side of the DNA nick is able to mediate a nucleophilic attack on the AMP enzyme complex, causing the AMP to transfer to the 5' phosphate position of the DNA. The AMP serves as a good leaving group for the nucleophilic attack of the upstream 3' OH, with the 5' phosphate to seal the DNA backbone and release the AMP. That is an interesting figure here for you to take a screenshot of and understand the entire aspect of the protein family name and the PFAM domain architecture, size of protein number of amino acids. Take screenshots and save them for future references. This is another one, which is the 3 consecutive nucleotidal transfers in the mechanism of DNA ligase, catalytic lysine numbering for ASFP ligase based on sequence alignment, adenylated enzyme is the product of transfer 1, adenylated DNA is the product of transfer 2, and ligated DNA is the product of transfer 3. Nicked DNA is bound subsequent to enzyme adenylation, adenylation. There's an LY extra I figured out. Anyway, beta B indicates nucleotide base. DNA ligase reaction. DNA ligase catalyze the crucial step of joining breaks and duplex DNA during DNA repair, replication, and recombination, and require either adenosine triphosphate ATP or nicotinamide adenine dinucleotide NAD+ as a cofactor. So shown on the upper left is the DNA ligase 1 repairing chromosomal damage. The 3 visible protein structures. Let's go back and just say shown on the upper left. It's the DNA ligase 1 repairing. So yeah, this aspect, okay? Right? The 3 visible protein structures are the DNA binding protein DBD, which is bound to the DNA minor groove, both upstream and downstream of the damaged area. The OB fold domain, OBD, unwinds the DNA slightly over a span of 6 base pairs and is generally involved in nucleic acid binding. The adenylation domain, ADD, contains enzymatically active residues that join the broken nucleotides together by catalyzing the formation of a phosphodiester bond between a phosphate and hydroxyl group. It is likely that all mammalian DNA ligases 1, 3, and 4 have a similar ring-shaped architecture and are able to recognize DNA in a similar manner. The upper right diagram is a high-resolution structure of E. coli ligin A in complex with nicked adenylated DNA from PDB to OWO, visualized by UCSF chimera. The various domains are indicated by different colors and relate to PFAM domains indicated. The lower diagram depicts the catalytic mechanism of DNA ligase 1. The ATP cofactor forms a covalent linkage with a lysine residue at the alpha phosphate position, casing the release of a diphosphate. The AMP is used to activate the 5' phosphate group, enabling the upstream 3' OH group to mediate attack in the central phosphorus atom. AMP serves as the leaving group. This is the video. Well, let me see if I can play it for you. Right.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called Helicase and results in the formation of a replication fork. The Separated strands each provide a template for creating a new strand of DNA. An enzyme called Primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
So I guess you had a wonderful video there with the overview of the DNA replication process, right? And we proceed with the subject matter. From there, now we move on to topoisomerase enzymes. Unwinding of the double-stranded helix at the replication fork generates winding tension in the DNA in the form of positive supercoils upstream of the replication fork. Enzymes called topoisomerases counteract this by introducing negative supercoils into DNA in order to relieve the stress in the helical molecule during replication. There are 4 known topoisomerase enzymes found in the E. coli that fall into 2 major classes: type 1 isomerases, type 2 topoisomerases. Let me repeat that: type 1 topoisomerases and type 2 topoisomerases, figures 9.10. Topoisomerase 1 and 3 are type 1 topoisomerases, whereas DNA gyrase and topoisomerase 4 are type 3 topoisomerases. Check these diagrams, take screenshots, and make notes, please. Structures of type 1 and type 2 topoisomerase: A. Crystal structure of type 1 topoisomerase, shown in blue, bound to DNA, shown in orange and yellow. B. Crystal structure of a type 2 topoisomerase. Topoisomerase 2 forms a tetrameric structure, shown in green and blue. ADP cofactors, pink, are shown bound in the enzyme. Now, type 1 topoisomerases relieve tension caused during the unwinding and unwinding of DNA. One way that they can do this is by making a cut or nick in one strand of the DNA double helix. The 5' phosphoryl side of the nicked DNA strand remains covalently bound to the enzyme at a tyrosine residue, while the 3' end is held non-covalently by the enzyme. The type 1 topoisomerases rotate or spin the 3' end of the DNA around the intact DNA strand. This releases overwinding in the DNA and effectively releases tension. The enzyme completes the reaction by resealing the phosphodiester backbone or ligating the broken strand back together. Overall, only one strand of DNA is broken, during the reaction mechanism. There is no requirement of ATP during the reaction. The E. coli topo-1 enzyme can only remove the negative DNA supercoils, but not positive ones. Thus, this enzyme is not involved in relieving the positive supercoiling caused by DNA helicase during replication. This is in contrast to eukaryotic topo-1 that can relieve both positive and negative supercoiling. Although E. coli topoisomerase 1 is not directly involved in relieving the tension caused by DNA replication, it is essential for E. coli viability. It is thought to help balance the reactions of the type 2 topoisomerases and help maintain optimal supercoiling density within the chromosomal DNA. Thus, topo-1 is thought to help maintain homeostatic balance of chromosome supercoiling within E. coli. Topo-3, which is also a type 1 topoisomerase, appears to play a role in the decatenation of the daughter chromosomes during DNA replication, but does not play a role in the relaxing of supercoiling. Note these diagrams, and let me read that explanation for you. Reaction of type 1 topoisomerases. During the reaction, type 1 topoisomerases nick one strand of the DNA. One end of the nicked DNA is covalently attached to the enzyme, shown in light green in the lower diagrams. The other end is held non-covalently and rotated around the double helix to unwind the supercoiling and relax the DNA. Once supercoiling tension is released, the backbone is resealed and the topoisomerase 1 enzyme is released. Topoisomerase, that is type 2, have multiple functions within the cell. They can increase or decrease winding tensions within the DNA, or they can unknot or decatenate DNA that has become tangled with another strand. It does so by a more dangerous method than their type 1 counterparts, by breaking down both strands of the DNA during their reaction mechanism. The enzyme is covalently attached to both broken sides. While the other DNA helix is passed through the break, the double strand break is then released. Note these figures. And the reaction of type 2 topoisomerases. The proposed type 2 topoisomerase reaction cycle is exemplified by topoisomerase 4. Topoisomerase 4 subunits are denoted in gray, cyan, and yellow. The gate or gDNA is in green, and the transported or tDNA is in mauve. ATP bound to the ATPase domains is denoted by a red dot. In step 1, the gDNA binds with the enzyme. ATP and the tDNA segment associate with the enzyme in step 2. In step 3, the gDNA is cleaved and the tDNA is passed through the break. Drug targetable domains within the type 2 topoisomerase complex are highlighted in subsections A, B, and C, with examples on the right-hand side of the figure. DNA gyrase is the type 2 topoisomerase enzyme that is primarily involved in relieving positive supercoiling tension that results due to the helicase unwinding at the replication fork. Type 2 topoisomerases, especially topo-4, also address a key mechanistic challenge that faces the bacterial replisome during termination of DNA replication. The circular nature of the bacterial chromosome dictates that a pair of replisomes that initiate from a single origin of replication will eventually converge on each other in a head-to-head orientation. Positive supercoiling accumulates between the two replisomes as they converge, but the activity of DNA gyrase, which normally removes positive supercoils, becomes limited by the decreasing amount of template DNA available. Instead, supercoils may diffuse behind the replisomes, forming precatenanes between newly replicated DNA. In E. coli, these must be resolved by topo-4 for chromosome segregation to occur. Thus, proteins and the termination of replication. Proper termination of DNA replication is important for genome stability. E. coli replication terminates in the region opposite, or IC. There are 1023 BP termination terse sites in the region, with some sequence variations that determine the binding affinities for the monomeric termination protein TUS. TUS binds to TER with high affinity in a 1:1 ratio, and TUS-TER can further form a very stable lock complex. F cytosine 6 of the strictly conserved G66 base pair of TER is flipped out of the DNA in duplex and bound in a preformed cytosine binding pocket of TUS, which shown in figure 9.13b. The TUS-Sturlog complex is polar with a permissive phase that allows the replosome to pass unhindered and a non-permissive phase that can block the replosome. The 10 TER sites are organized as two oppositely oriented groups of 5, allowing the replosome to pass the first group and be blocked to the second. This ensures that the two replication forks converge in the terminus regions for proper chromosome segregation. So check this figure out, take screenshots please, and make notes. They could be very helpful. And note the details as I read out this as well for you. Figure 9.13, mechanism of replosome blockage by TUS-TER replication termination complexes. A. Schematic representation of the E. coli chromosomes showing positions of RIC and TER sites. The clockwise moving fork passes through these permissive sites shown in green and is rested at the non-permissive sites shown in red. B. Schematic representation of TUS of structure of the locked TUS-TER complex PDP 2106 showing cytosine 6 in its binding pocket in TUS. C. Interactions of residue arginine 198 of TUS with both strands of TER in complexes with double-stranded wild-type TER PDB 2105 left and the TUS-TER UGLC complex GC6 base pair inverted PDB 4x4R3 right. Alright, we go to the next topic of 9.3, that's DNA replication of extrachromosomal elements, plasmids and viruses. To copy the nucleic acids, plasmids and viruses require frequently used variations on the pattern of DNA replication described for prokaryote genomes. So we got to focus on the style known as the rolling circle method. So rolling circle replication. Whereas many bacterial plasmids replicate by a process similar to that used to copy the bacterial chromosome, other plasmids, several bacteriophages, and some viruses of eukaryotes use a rolling circle replication. The circular nature of plasmids and the circularization of some viral genomes on infection make this possible. Rolling circle replication begins with the enzyme nicking one strand of the double-stranded circular molecule at the double-stranded origin DSO site. In bacteria, DNA polymerase 3 binds to the 3-OH group of the nicked strand, begins to unidirectionally replicate the DNA using the the unnicked strand to act as a template, displacing the nicked strand as it does so. Completion of DNA replication at the site of the original nick results in a full displacement of the nicked strand, which may then circularize into a single-stranded DNA molecule. RNA primase then synthesizes a primer to initiate DNA replication at the single-stranded origin SSO site of the single-stranded DNA ssDNA molecule, resulting in double-stranded DNA dsDNA molecule identical to the other circular DNA molecule. So this is a figure on that. SSO DSO to a nick at 3' to in the presence of DNA polymerase 3, we get displacement of next strand towards the DNA ligase, dsDNA synthesis of first strand, and the ssDNA synthesis of the second strand. Figure 9.14. Rolling circle replication. The process of rolling circle replication is initiated by a single-stranded nick in the DNA. Within prokaryotes, DNA polymerase 3 is utilized to generate the daughter strand. DNA ligase rejoins nicks in the backbone and enables the initiation of DNA synthesis of the second daughter strand. The next one will be 9.4, that's DNA replication in eukaryotes, the cell cycle, which we just discussed in the last lecture. Okay, coming to the cell cycle part of the DNA replication, we have the cell cycle is an ordered series of events involving cell growth and cell division that produces two new daughter cells. Cells on the path to cell division proceed through a series of precisely timed and carefully regulated stages of growth, DNA replication and division that produces two genetically identical cells. The cell cycle has two major phases, interphase and the mitotic phase. During interphase, the cell grows and DNA is replicated. During the mitotic phase, the replicated DNA and cytoplasmic contents are separated and the cell divides. So watch this video about the cell cycle at HTTP OpenStax.org. So let's just check that too.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting.
So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So, ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So, 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there, and it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells, and in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So, in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So I think that was a neat explanation about mitosis and meiosis. And close approximation are in fact a good reflection of the subject matter that I was discussing with you. So I guess that should be it. And we go into the next part of the subject matter proper. Right, so this illustration shows a cell cycle which consists of interphase and mitotic phase. Interphase is subdivided into G1, S and G2 phase. Cell growth occurs during G1 and G2, and DNA synthesis occurs during S. The mitotic phase consists of mitosis, in which the nucleochromatin is divided, and cytokinesis, in which the cytoplasm is divided, resulting in 2 daughter cells. And the diagram of the cell cycle, a cell moves through a series of phases in an orderly manner. During interphase, G1 involves cell growth and protein synthesis. The S phase involves DNA replication and the replication of the centrosome. And G2 involves further growth and protein synthesis. The mitotic phase follows interphase. Mitosis is nuclear division, during which duplicated chromosomes are segregated and distributed into daughter nuclei. Usually the cell would divide after mitosis in a process called cytokinesis, which the cytoplasm is divided and two daughter cells are formed. During interphase, the cell undergoes normal processes while also preparing for cell division. For a cell to move from interphase to mitotic phase, many internal and external conditions must be met. The three stages of interphase are called G1, S1, S, and G2. The first stage of interphase is called the G1 phase, or first gap, because little change is visible. However, during G1 stage, the cell is quite active at the biochemical level. Next lecture.